Good afternoon. Right. In the previous we have discussed regarding what? The photoperiodism, right? So photoperiodism means what? It is the duration of exposure to the light which induces flowering. We know that the hypothetical hormone called florigen, which is making the shoot system to be get modified into the reproductive effects. The shoot effects ಏನಾಗಿದೆ? Reproductive effects ಆಗಿ modify ಆಗಿದೆ. That is what? Due to the activity of light. But not all the plants in the plant kingdom are dependent on the light to induce the flowering in angiosperms. Some plants which are dependent on day length, some plants which are dependent on the dark length, that is the night length. How? That is what? The short day plants, long day plants, and some are not correlated with the light or darkness period. They are day neutral plants. So there is a critical period of light for every kind of the plant. It varies with the plant types. For example, if it is for a cereals, it is around 11 hours of a photo period. There may be other dicotyledon plants which are having the 14 hours of the day length. That is what? It may varies, means it always varies with the type of the plant. That is what? But here, the critical period, what we are saying, above that the light is required for inducing the flowering, then it is called what? Long day plants. The example we quoted here is what? Rhizome, example, there is the chrysanthemum, xanthium, and then strawberry. These three we have quoted that for the long day plants. But there is no short day plants, okay? But there is no examples that are prescribed in the NCERT, but this for the short day plants. But what is the short day plant then? They require less photo period than the critical period. If the critical period is 11 hours, then they require less than the 11 hour, not equal also. If they are equal, they will not induce the flowering. If it is more than, they will not induce the flowering. If it is interrupted, that is also not giving the flowering. That is what? They need a continuous exposure. That is what we have discussed the different cases with the diagrammatic representation. So this is what we have studied with respect of that. But as I said, there are some plants which are not correlated with the photoperiod exposure. That is what? Light or darkness exposure. What is that? That is the day neutral plants. Example we can see is tomato. That is for flowering. But the tomato seed will not get germinate unless it exposed to the light. But I am saying only for initiating the flower. The photoperiodism is mainly for inducing flowering. Okay. So there is one of the extrinsic factor as we studied with the light. But there is another one extrinsic factor called temperature, which is mainly required for inducing the flowering. That is what? Vernalization. What do you mean by vernalization? The vernalization is nothing but the treatment of temperature at a low condition. That is what? The low temperature treatment. That is about the 5 degrees Celsius. Up to 5 degrees Celsius we can minimize the temperature of the plant body, because we know that the optimum temperature is about 25 to 30 degrees Celsius. But we can minimize that one to avoid what? Precocious maturity. What do you mean by precocious maturity? That is early maturity. So what happens to that one means when the precocious maturity is attained in the plant body, it may not get a complete vegetative structures, especially the leafy vegetables. Means the vegetative structures are edible, like the cabbage or lettuce, right? Or the carrot, beet. So these are all vegetative structures which are edible, and that content is required more. Instead of the flowering in the carrot, instead of the flowering in the cabbage. That's why the thing here is they are avoiding the precocious maturity to give sufficient duration for the vegetative development. So that is what? The vernalization. So vernalization is what? It is treatment with cold temperature. The treatment of plants treated with this one, the cold temperature. Okay. So for what? Avoiding the precocious maturity, that will give the sufficient duration for getting a vegetative structures. But there are different kinds. because this vernization is affecting the different kinds. Okay, the vernization, how it affects? There are annual plants, there are biennial plants. Okay, that is different for those kinds. For example, if you get through the winter varieties like the barley, wheat or oat, these varieties, because we know that here if these varieties, if they are spring, means there are some spring varieties, because we can find the two types of these varieties. Some of them are maybe spring varieties, some of them are maybe the winter varieties. In the usually winter varieties are what? They are cereal plants. Usually winter season ನಾವು sow ಮಾಡ್ತಕ್ಕಂಥದ್ದು ಯಾವುದು? They are cereal plants. That's why in the winter condition we can sow the cereals. Winter plants means the cereal plants. Okay. ಹಾಗಾದ್ರೆ the spring season which is between what? So winter and summer, they will alternate the two different seasons. What do you see? Autumn and spring. So spring season is always after summer, before winter. So after the summer, before the winter, the season is called autumn. Then the spring season is after winter, before summer. Clear? So after winter andre yavadu? Nanu nimage ಹೇಳ್ತಾ ಬಂದೆ. So this is in commonly what we call ಹೆಸರಲ್ಲ, ಶರದ್ ಋತು, ವಸಂತ ಋತು. ಅದಕ್ಕೆ ನಾವು ಕನ್ನಡದಲ್ಲಿ ಸಾಂಗ್ ಹೇಳ್ಬೇಕಾದ್ರೆ ಹೇಳಿದ್ವಲ್ಲ, ವಸಂತ ಕಾಲ ಬಂದಾಗ ಮಾವು ಚಿಗುರಲೇಬೇಕು, ಕೋಗಿಲೆ. ಯಾಕೆ? The tips are growing very short at the winter season, right? So here we can see the germination of seeds, but they will attain very less growth in the winter season. But they will get complete yield in the early spring to late spring, or nothing but up to the midsummer they will attain their reproductive stages and they will give the yield. That is what the condition actually. So coming to the NCERT point, if we sow the spring variety, if we are sowing the spring variety in the spring season, it will give the yield within the completion of the season. Our season complete agodrolage it will give the flower. Means if we sow the spring varieties in the spring season only, the sowing of spring variety in the spring season will give the yield before its end of the flowering season. If means that means before the ending of spring, that is what late spring, nothing but early summer. Before that summer only it will give the yield. But the condition is, if the winter varieties are sown in the spring, if the winter varieties are sown in the spring, it will not initiate the flowers in the spring season or the summer season. Why? That is what the thing we are saying, that there is a particular temperature which is required to initiate the flowering in the plant bodies. That is what I said. There are two varieties. One is winter variety, other one is spring variety. Clear? So they have, in the NCERT they have clearly mentioned the barley, oat and wheat are the three different plants we can see. They are of two different types. One is the winter variety, another one is a spring variety. If the spring variety is sown in the spring season, it will give the yield before ending of its season. But if the winter variety is sown in the spring season, it will not give rise to the flowering before end of that season. Hence, it is always preferred that the winter varieties need to be sown in the autumn season. What happens in the autumn season? We know that autumn means where we can see the fall of the old leaves and arise of the new structures. The bud will be arising here. The new initiating structures will be formed here. That's why here when the winter variety is sown in the autumn season, the seed will get germinated. The seed will get germinated. So this seed will develop very minimum structures in the winter. After that treatment, means in the winter when they get exposed with the lower temperatures up to the up to 5 degrees Celsius, means less than 30 up to 5 degrees Celsius, we can see that condition in the winter varieties. When they expose us to that kind of low temperature, that is what the cold treatment, they will initiate their more vigorous growth in the early spring to the late spring. And before the summer, that is means end of the summer, that is at the mid summer, they will give the yield only after proper treatment. Hence, there is a clear mention in the last line of that one, last paragraph. A particular plant is giving a flowering means there is a particular temperature which is making the photoperiodism in stimulus. The photoperiodism is stimulated means easily they will give what? Flower. So that's what in the biennial plant. The another next condition is in the biennial plants, like which are the biennial plants? They complete their life cycle in the 2 years, right? But here they have mentioned they are monocarpic in condition. What are those? They are monocarpic in condition. Mono means what? They are giving only the flowers in their lifetime only one time, even though they may be perennial, like the bamboo. Once, sorry, not bamboo, like Neel Kurunji, or once in a 12 years. But the thing is, but if they complete their life cycle after their flowering season or after the flowering, then they are called monocarpic condition. Mono means in their lifetime they will flowers only once, whatever may be their lifetime, whether it may be 3 months, whether it may be 3 years, whether it may be 30 years or 300 years, whatever, but they should only end their life by flowering, one time flowering, that's it. So mono refers to single, carpic refers to carpel, nothing but gynoecium, means the flower structure. So here the monocarpic biennial plants like they need what? A particular low temperature. So particular low temperature is required them. Why? To give the stimulus for photoperiodism. That is what initially I quoted. The vernalization is different in different kind of plants. As we have seen the annual plants, now we are seeing the biennial monocarpic plants. And as we have avoided the precocious development, here we are making the biennial plants to get early maturity, means once its vegetative structures are get completely developed, it initiates its flowering. That means it will be ending its vegetative growth. Clear? That is what in the biennial plants, like the monocarpic varieties like radish, carrot or beet or cabbages, here the vegetative structures are edible. Illelladrallu kuda reproductive structure is edible idya? No, all these are having the vegetative structures are edible in these varieties. Hence, at certain temperature of exposing, means not the regular 30 degree or 25 degrees Celsius, minimize the temperature, that low temperature will give the stimulus, along with that it will initiate the flowering with the photoperiodism. That's why there is also an hypothetical hormone called vernalin. As we said about the florigin, right? How? The florigin, as we said, it is synthesized by the phytochrome which is present at the site of light perception in the photoperiodism. Then what are the site of vernalization? Which structures in the plant body are the sites for getting the low temperature absorption? That is what? Shoot, apex, or it may be the embryo present in the seed. You got the point? The site of perception for vernalization, that is very important, because the photoperiodism, the mature leaves, okay, because completely development of what? The chlorophyll pigment, that is the chromoprotein here we are saying, that is phytochrome, that is present in the mature leaves only, right? That's why the site of perception for photoperiodism is different than the site of perception in the vernalization, because the site of perception for cold treatment is either embryo present in the seed, okay, or the shoot apex. Clear? ಅರ್ಥ ಆಗ್ತಿದೆ ಎರಡಕ್ಕೂ ವ್ಯತ್ಯಾಸ? There we mentioned it is not shoot apex, it is only leaves. That is in photoperiodism. Here it is not leaf, it is shoot apex and also embryo which is present in the seed. Very important site of perception in vernalization and in photoperiodism. Clear? So this is all about vernalization. Clear? So one is photoperiodism and the one is vernalization. These two are what? The extrinsic factors which are making the flowering. That is, which are affecting the maturation, which are giving rise to the reproductive maturity. That is what the plant development is dependent on these two and also other extrinsic factors. But we have detailed about two. One is light, another one is a temperature. But now we will move towards an intrinsic factor which is bringing a different condition the plant growth and development, especially the intrinsic factor which is making the seed to be remaining in an inactive state. What is that means? It will not get germinated, it will be not getting further development. The seed will remain as it is. What is that? That is the dormancy. The seed dormancy. What is seed dormancy? Seed dormancy is the seed will not get germinated even in spite of the presence of the favorable conditions. That is extrinsic factors all favorable idave, but the seed is not getting germinated. Why the seed is not getting germinated even in spite of the presence of all the favorable conditions like the water, oxygen, humidity, temperature? Right? That means the intrinsic factor like the genes which are making the seed to be get in a arrested state, that is a non-germinated condition, that is dormancy. So seed dormancy. What is seed dormancy? Seed will not germinated the seed will not get germinated in spite of the presence of all the favorable conditions like light, temperature, humidity, oxygen. Why? That is because of the seed will remain in a dormant stage because of intrinsic factor. That is, they have mentioned endogenous. Endo refers to inside, genus refers to the synthesis. They are endogenously affected. Okay, so that is what if the seed remains in an dormancy, that is means because of only the endogenous factors. What are the endogenous factors? That is what I mentioned, intrinsic, that is here the gene, or maybe due to the chemicals, okay, or maybe due to the physical barriers. But physical barrier is not intrinsic factor. Okay, but seed coat, that is physiologically which is making its testatagma to be get more harder and thicker one. Like this, the gene which is making the seed to be get in a dormant stage, or the chemical which are synthesized in the plant body or in the seed that is making to be in a dormant stage, or the physiological, they are making the special thickenings in the seed coat that is making the barrier. Right? So that is what these are the intrinsic factors. Then what are that? As I said, genes, and the chemical, that may be the hormones like abscisic acid or phenolic acid. This is very important. Abscisic acid, we already know that it is a dormancy bringing hormone, along with that the phenolic acid, right, and along with that the para ascorbic acid. The last line in the NCERT they have mentioned, okay, the para ascorbic acid, make it underline, okay. So these three are the chemicals which are bringing the dormancy. They are dormancy bringing hormones, okay. But even the gene which may the seed to be in a dormant conditions also, the genic expression, and physiologically how making thick development of seed coats. The oval integuments are get thicker one, that is testa and tegma are more thicker than the normal conditions or any other seeds, then it will also act as a seed to be remain in a dormant condition. That is also intrinsic one, okay. So the thick seed coat, clear. So these are the different conditions, but that is also due to the early embryo development. The seed is not getting germinated because the embryo is not completely developed. See, the wheat which is grown in the this year, we will not use those grains of the wheat or those cereals into the immediate next season sowing. Why? We will give the a proper period of rest, means the proper gap period for harvesting period to the next sowing period. Why? Means these grains will be not having a embryo which is completely matured one, which is not completely developed mature embryo is not present in that grains. Hence the duration is given before the sowing, after the harvesting, to get a complete embryo development. Even after harvesting, means the first harvesting to sowing we will give a gap. Why? Means to give the embryo a proper time to get the development, clear? So that is what the thing is. Hence if the early embryo is present, yes, the seed is matured, but the embryo is not completely developed. Hence for development of that embryo we will be giving a proper gap period. At that gap or that duration will take place the development of embryo. Hence we will use after 1 year or after one gap period we will use that seeds as sowing seeds, clear? That is what why we are avoiding immediate harvested grains to be sowing. We will give a gap period only to give the condition to avoid non-viability of the seeds, because if they are viable easily they will give rise to the radicle and plumule. The non-viability is because of immature embryo, improper development. These are all the different conditions. We will see the seed is in a dormancy condition. Then how to break the dormancy? We can break the dormancy by mechanically, we can break the dormancy by treatment with the chemicals, right? So or physical treatment with the any water or any other cold treatment, right? So how? That is what the dormancy bringing conditions are these all, right? How to break this dormancy? Let's see. We can make abrasions, either natural abrasions or mechanical abrasion. What is abrasion? The removal of the outer layer. Zoological skin disease is disorder of a skin, that is called abrasive skin. What is abrasive skin? The removal of outer layers. Yuvraj, outer layer remove ಆಗ್ತಾ ಇದೆ ಅಂತ ಅರ್ಥ, ಚರ್ಮ ಸುಲಿತಾ ಇದೆ ಅಂತ ಅರ್ಥ ಏನು? That is what the skin disorder called abrasion, clear? That means here we are making the seed coat by removing its outer coat. Testane ತೆಗಿಬೇಕು, ಟೆಗ್ಮನೆ ತೆಗಿಬೇಕು, outermost layer. Here the abrasion is made either mechanically by knives, okay, or by rubbing them against the sandpaper or even any hard surface, okay, or even by the vigorous shaking. These are the mechanical abrasion process. This vigorous shaking process is called impaction. What is impaction? The vigorous shaking of the sheets to remove the outer layer, that is to make the mechanical abrasion, that is impaction. Vigorous shaking is called impaction. Ee term nenpirlebeku. Okay, so impaction is nothing but vigorous shaking of sheets which leads to the last term kuda nodri, third line alla second line alla ideyale vigorous shaking. That vigorous shaking is the removal of outer layer of this anything, but abrasion andre here that is due to the impaction, that is means by vigorous shaking of the sheets we will be removing its outer layer, that is mechanical abrasion. Then how to make them naturally abrasive? The natural abrasion is the abrasion process by the natural one, that is by the animals. That is simply by the animals. How? Especially the strawberry seeds or the blackberry seeds, or what we will commonly refer that berries, not that type fruits. These strawberries and blackberries. Along with that, walnuts. Okay, the walnut will not, or any of the nuts what we will refer, these nut type fruits will not get germinated if there is no squirrel. There will be no germination of the nut fruits. The nut fruit is completely dependent on squirrel for its germination. The nuts are get germinated by the squirrel. How? Means they are helping, not the squirrel will make the germination, but squirrel is making the natural abrasion by its consumption. It will remain, so it will remove the outer layer, that is the naturally it is making the abrasion. It is not intention that is to make the germination of the nuts, but natural process of feeding, it will remove the outer coat of the seed, that is abrasion by the squirrel. Like that there are plenty of the birds which are related to this one. Okay, even the tomato seeds will not get germinated. Why? If they are not soaken, if they are not easily soaken in the intestinal juices, comparatively with the intestinal juice soaked tomato seeds and normal soaking seeds, normal seeds which are without the soaking in water also, we will not see the germination of the seeds. Why? The intestinal juices will make this tomato seed to be get easily germinated, but the tomato seed coat is highly impermeable. That's why it will not get germinated in the intestine itself. But why? Because the light is very important to induce the germination in tomato. That is special case. Without the light, the tomato seed will not get germinated. You can check that one. If they get the minimum amount of the light induction, then only they will give the germinating, that is what germinated conditions. Clear? So that's why not only the squirrel, there are different kinds of animals, they are involved in that one, especially when they are passing through the animal gut region, or when they come with the different enzymatic actions of the digestion processes or digestive tract, that is gut. They will come with the contact of enzymes. That enzymatic action will break the abrasion, means break the seed coat layer. That is what? The natural abrasion. Mechanically and naturally. This mechanical is what? Physical. Physically we are removing the coat. Okay. There is a process called this removing by the knife or the sandpaper. The seed coat is making its mark. This process is called scarification. This is very important, repeatedly asked content. Scarification is the main process which brings the abrasion and makes the seed to be get germinated without any barriers. That makes it more permeable. That is scarification. Other thing is impaction. That is not artificially we are doing. But naturally, this is the intestinal juices or the juices or enzymes which are present in the digestion process will make the seed coat to be get removed. Especially the animals which are involved, like the nuts will get germinated by squirrel. Clear? And by the chemical how we can make that one? Means by the chemical means one is the gibberellic acid along with the nitrates. The nitrates and gibberellic acid spray or the treatment with the nitrates and gibberellic acid will make the seed to be get germinated. That is why it is called anti-dormancy hormone. Clear? It is showing an antagonistic property with the abscisic acid. Clear? So even also by the cold treatment to the some seeds, we can make the seed to be get germinated. That is also the other condition. Clear? So likewise, the light temperature also making the seed to be get germinated by induction. Treating with them of particular temperature make them to be get germinated. So this is all about the seed dormancy and breaking its seed dormancy. Once the seed is get broken its dormancy, it will start to develop the radicle. That radicle will be differentiated into radicle and plumule. Then that plumule will give rise to the shoot system. Radicle will give rise to the root system. But here once it is based on the hypocotyl region, that will give rise to the cotyledon above the land surface. If the cotyledons comes above the land surface, then it is called epigeal germination. It is extra I am saying. Okay? And if the cotyledons are remains inside the soil surface, they are hypogeal. If it is a land surface, the cotyledons are, these are cotyledons what I have shown here. Okay? These are axis. Right? The cotyledons are comes above the soil surface. But in some condition, the cotyledons will remain inside the soil surface. They will not come out. Okay. So here, the tamarind we can take. Okay. Tamarind. Right. So that will, the cotyledons will come above the soil surface. That is why it is called epigeo. That is what? Above the soil. The cotyledons will come. That is epigeal germination. If the cotyledons will remain inside the soil, that is hypogeal germination. This is how the hypocotyl region is acting. Whether it is pushing the cotyledons above or whether it is pushing them inside the soil. The hypocotyl region makes the major action here in the bringing cotyledons above the surface or making them to be remain inside the soil. That is what epigeal and hypogeal. Can you remember the example for epigeal? Okay, tamarind. Then any other? Even the grams, they all comes above the surface. The cotyledons will be first thing. Okay, that is what here we can quote. Then hypogeal, groundnut, best example. Right? Mango, mango seed, cotyledons will come above the surface. What am I barata? Illa. Will mango give the cotyledons above the soil surface? No. Mango, these two, groundnut, these are hypogeal. Mango and groundnut, hypogeal. Then what about the paddy? What about paddy? Hypo. The seeds will not come over the surface. Means that cotyledons will not come over the surface. No. What about jowar? What about maize? The cereals will remain in the soil itself. Okay. Their cotyledons are only one. Right? Okay. Then if the seed is, means the seed coat will come above the surface and cotyledons will be expanded, then it is epigeal. Just you can remember the grams. Okay. Clear? So this is all about the germination. But in the pea seed, as we mentioned, the while germination, the seed coat is broken due to all these factors, intrinsic and extrinsic, all the thing is favorable, then seed coat is removed and it will give us the hypocotyl, but the hypocotyl region will comes as a hook-like structure. This is the characteristic feature of pea seed germination. Idu kuda kelidare onsala nenpalli. Okay, so this is all regarding seed germination. Clear? So in this chapter we have started with the seed to its germination and then elongation, maturation, that is by the differentiation, and then it will be get made dedifferentiated and again redifferentiated, or it may face the senescence and due to that may end the life by death. So that is what all the changes with respect to of its birth to death during its growth and differentiation, that is what the development. This is all about the chapter called plant growth and development. Is it clear? Okay, thank you.